Congress again.
Welcome to the clinical research tour. We're going to wait just a minute to get started. We have a lot of people that are going to enter into the seminar, to the webinar, so we'll start in just a second. Welcome to the Clinical Research Virtual Tour. We're really excited that you came. We're going to wait just a minute to get started. Make sure everybody can get in and get their audio settings fixed up. Nice to see a lot of familiar faces in the attendee box. Glad that so many of you are able to join so many of our events for the virtual conference. All right, we have quite a few people still coming in, so we're going to wait just a minute and then we're going to get started. Okay, we are going to get started with our virtual tour. My name is Michelle Dvorak. I am your program service coordinator here in San Antonio, Texas at the Chromosome 18 Registry and Research Society. And we are super excited to share our first ever clinical research virtual tour. And you will be meeting Dr. Cody in just a minute. She will be taking us on that tour. But first, I had just a couple of announcements before we get started. Wanted to remind you of our online auction that is going on. Unlike in person when we have conference, when we do it in person, we don't bid the whole way through. But for our virtual conference, you can bid now. You can bid today. As soon as this tour is over, you can take a visit to the online auction site and you can make bids there. We have some wonderful merchandise there for you to bid on. And today, while we're on the tour, feel free to put questions in the chat box. If it's a question we can answer right then, we'll go ahead and shout it out to Dr. Cody. If it's something that we'll be addressing at the end, then we'll save it for then. But go ahead and write it in there as soon as you have the question, put it in the chat box. So, well, without further ado, I am going to turn it over to our wonderful Executive Director, Neil Parker.
Thank you, Michelle, and welcome everybody. This is actually a very exciting day for us that we are providing our first virtual tour. To give you a little bit of context is that you would have kind of come up in the elevator to the 5th floor, and we are situated in the pediatric department of UT Health San Antonio. You would have walked down a long corridor to come to the area that the Chromosome 18 Clinical Research Center has been for almost 30 years, actually next year. Being part of UT Health is incredibly important to us because of the collaboration with so many other wonderful physicians, doctors, and also all the staff that have helped us out in so many different areas. So for people that don't know Dr. Cody, I normally kind of tell a little bit about the story about when Elizabeth was born 35 years ago, that Janine went back to school, got her doctorate, and started the Clinical Research Center. And as I say, we're going to be celebrating 30 years of that next year. So kind of like a huge achievement. And Dr. Cody has been leading the charge for all of these years, and we have a tremendous debt that I think every Chromosome 18 family doesn't realize kind of how much Janine has done for everybody. And on that note, I'm going to pass it over to Dr. Janine Cody, who's going to carry on with the rest of the tour.
Okay, I got to take that off so you can hear me. Thank you for joining us. As Neil said, we're on the 5th floor of the medical school at UT Health and the Department of Pediatrics. And so this is what you would see when you first come on the tour. We want to make this feel like a tour, so we're trying to move you along through as you would if you were really here. When you first get here, here's our bulletin board that Anna's created, one of 5, with all of our kids. The families send pictures that are intended, they send them knowing that they're going to go up on a bulletin board. So everybody gets a chance to see all these amazing smiles. So here and as Neil said, if you have a question as we go, please don't hesitate to ask. And if it's something, type it in the chat box. If it's something that I need to answer right away, Michelle will chime in. If it's something we'll get to later and she knows that, then she'll hold the question. So here, let me go over just a minute the conditions. Whoops, we're having a camera issue here. Okay, are you ready? Just to make sure everybody is aware, interested in, there are 5 groups of conditions. And people with 18q minus, they have 1 normal chromosome 18, and the other chromosome 18 has a missing piece of any size from any position somewhere in the long arm. The long arm is called the Q arm and the short arm is called the P arm. So as you can imagine, there could be a lot of variety, and there is a lot of variety in people with 18q deletions. Likewise, you could have a missing piece from 1 copy of the short arm, the P arm, and again, that missing piece could be in any position and of any size on the P arm, and that's 18p minus. Then there's ring 18, in which one of the chromosome 18s has missing pieces from both ends of the chromosome, and the chromosome forms a ring. In order to form a ring, the caps that are normally on the ends of the chromosome both need to be missing. And so by definition, you have a missing piece from both the P arm and the Q arm. And then there's, of course, trisomy 18, which is by far the most common, in which individuals have 3 copies of chromosome 18. And before we existed, there were groups and researchers interested in trisomy 18. And so we've not really focused on that as much. It's also much more common. So our interest in trisomy 18 has really been on the long-term survivors, those over 4 years of age. And 18q minus and 18p minus occur in about 1 in 55,000 births. But the most rare, 1 in 600,000 births, is tetrasomy 18p, in which individuals have 2 normal copies of chromosome 18, but then they have a 3rd chromosome 18, but it's made up of 2 P arms. So there's 1, 2, 3, 4 P arms. That's tetrasomy 18p. And we enrolled people from all over the world. This is a map Anna's created from showing where everyone lives. The limiting factors, you can see they're mostly from North America. The limiting factor is really the ability to speak English and to send us blood that can get here in just 2 days. We could always translate our consent forms into other languages and enroll people in other languages, except one of the key aspects to what we do is the fact that this is a longitudinal study. We collect medical records ongoing. We keep asking families to send new medical records as their child gets older and has more information or more conditions develop or more evaluations are done. We also send out a lot of surveys. And so all of that would have to also be translated. So, and many of the instruments, especially for behavior, are all normed for English speakers. So we're really limited because of all those reasons to people who can understand English, where it's people that are fluent in English. But you can see we have families enrolled. We have almost 700 families enrolled at this point, and they come from all over the world. And some of them actually, the families in Mexico, we can't get blood from Mexico, so those families, when they visit the States, they arrange to have blood drawn and sent to us when they're visiting. And here we have the team of researchers, myself, Dr. Hale, who serves as the medical director, although he's moved to Hershey, Pennsylvania. Here are our research staff, and Annis Hill, I think everybody knows, is project manager. You meet her in a second. And Mimi Hassi is a research associate. Mimi actually went to medical school in Kosovo, but is not licensed to practice in the United States, but she has all that medical knowledge. And so what she does is she goes through everybody's medical records and pulls out the key features and the important things and monitors the medical records and gets those all into our database. And also when we have studies where families come to San Antonio for evaluation, she accompanies them to each visit to make sure they find the right place at the right time and no billing information is obtained from the families, that sort of thing. And Mimi and Annis are actually our newest members of our team. They've only been with us 13 years. So we have lots and lots of work experience that brings to bear. This is Bridget Swallow. She is our psychometrist. She has a master's degree in school psychology. So she's the one who sends out most of the behavioral surveys. So lots of families communicate with Bridget as well as Annis. And Patty Hurd runs the lab. You'll meet her in just a few minutes. Courtney Siebold is our genetic counselor. She actually lives in Germany at the moment. She was here for a couple of years and her husband's in the Air Force, and so then they moved to Russia for a couple of years, and then Germany, and then Ohio, and then back to Germany. And since our families are all over the world, most of our communication happens by email, and so people probably don't even know she's really in Germany. And then we have David Rupert, who's our data manager, helps with the database, and is also our photographer. So if you don't like this, it's David you can blame for their, stop it, David. Dana Word is a research nurse, and then we have administrative staff, Gloria Matthews and Linda and Sarah, who keep track of us and budgets and that sort of thing. And then we have all our listed collaborators here. These people, as I think you all know, since there's 263 genes on chromosome 18, pretty much every body system could be affected. They, in any one person, of course, they don't have all the various potential conditions. But if we're going to assess a group of people, we need to have all kinds of medical specialists. So we have neurology and immunology and neuroradiology. We also have some basic science collaborators who are specialists in mouse behavior, mouse neurophysiology. And there was a time when the Chromosome 18 Clinical Research Center paid portions, maybe 10% of a lot of these people's salary. And so when we had data that needed to be analyzed, they analyzed it. We had papers that needed to be finished up. They got it done because they had this time that was protected that was for us. We no longer have funding able to cover anybody else's salary. And so they're all volunteers at the moment, and they all have lots of other responsibilities. And so it makes getting some of what we need done slow, and it turns my job into lots of harassment. So, but we have many of these people you've met, many of these people you've probably heard their talk at the conference, our virtual conference this year. And over time, as we've learned more about the chromosome 18 abnormalities, some of the specialists are even more specialized, like people specializing in platelet function and bone health, for example. So let me know, are there any questions, Michelle, that need to stop at this point?
We do have a question, but it's one that'll be better for the lab.
Oh, okay, that sounds good. Well then, here's Annis. Annis can wave. Here's our office suite where I live, where Annis lives. Everybody already knows Annis. Mimi's around the corner in here. And across the hall are David's office and our administrative offices. And then we're going to go down the hall and around the corner and to the lab.
And whilst Dr. Cody and David are moving along, just wanted to say a couple of things. Is that for people that don't realize is that, you know, the Chromosome 18 Clinical Research Center is the only dedicated research center in the world. And a lot of the information that's out there kind of on the internet that you might, when you first get your diagnosis, look for, you know, a lot of the valid information has kind of come from the Clinical Research Center. So just a thing to remind everybody. It looks like Dr. Cody's walking into the research center, so let's go back to her.
Dr. Cody, can you repeat your introduction to come into the lab? We didn't hear that part.
I didn't say anything.
Okay, it looked like you might have been saying something. We just wanted to make sure we didn't miss anything.
So I was just directing David to point to that there's two labs. The first lab, and then this is our small tissue culture lab. And this is Patty Hurd, who I sort of introduced you to by picture before. She runs the lab, and she's going to take over from here. Hello, everybody. If you have questions, go ahead and do what you're supposed to do, which I guess is type them, and someone will interrupt me. That's okay. Worst that'll happen if I lose my train of thought, and I'll ramble. So, I'm Patty. I'm the lab technician for the Chromosome 18 Research Center. I'm the person that receives your blood and processes it. So I do a few things with the blood. First, if you have sent blood before, you know that you ideally have sent a small green top tube. I set up a culture that has to incubate for 3 days, which is why if you're international, we only want the blood. Oh, sorry, David's cueing me to speak louder. We want the blood shipped on a Monday, so I have plenty of time to set stuff up. And with that, I'm setting up a prep so that we can visualize the chromosomes under a microscope. Normally, that's a little difficult to do, so I add chemicals, get them nice and condensed, and then we can stain them with specific probes in a process called fluorescent in situ hybridization, and we will get images that look like this. I apologize because it's kind of blurry, but when I zoomed in, that happened. So these are the chromosomes from one cell. I have probed or added 2 regions of DNA that are going to go find their matching counterpart on the chromosomes. The green dot here is looking for the centromere of chromosome 18. It's specific for 18. The centromere is that, I think Dr. Cody calls it a belt? Waistband. Waistband. And you got to have the waistband to have a chromosome. So you'll see, here's one lighting up on this chromosome. And here's another green dot lining up there. So we know there are 2 chromosome 18s. Then I added a probe that's looking for the end of the Q arm, the telomere, and it's labeled red so we can tell them apart. And it's only showing this, it only lights up on this chromosome. So we know this chromosome is deleted for that reason. And what's nice about this, and I'm going to come back to the computer and show you in a second, is this lets us see the shape of the chromosomes. And so a deletion is kind of straightforward, but if we have a duplication, we don't know where that extra material is. It could be stuck on the end of another chromosome. It could be stuck on 18. So by doing this technique, we can actually visualize the chromosome. So one of the other things I do with the yellow top tube is extract DNA directly from the blood. It's not a very complicated procedure. It takes a couple hours and then it's overnight incubation, rocking at room temperature to go into solution. Because the second to the last step, and we'll have to work with me here. Am I holding it in the right place, David? Yes. I don't know if you guys can see.
We're able to see it.
There is
what looks like thread floating in here, and it is not thread, I promise you. It's actually my DNA. So when Dr. Cody wants to clone me, here it is. Me and Tyrannosaurus Rex.
It could be a fundraiser. Paul will pay me not to do that.
Yeah, I don't know if you heard that. He said my husband would donate to the registry to not have me cloned. So this is not usable in this form. I would have to take off this liquid, add a solution so if the DNA dissolves, and then it's ready to go. And what do I want to do with the DNA? I want to figure out what areas of chromosome 18 are duplicated or deleted. So I use a slide. These are actually usually kind of inexpensive. This slide, the prices have gone up, is about $1,200. And it's called a microarray slide. And what it has on it are 4 identical regions that each have 180,000 pieces of DNA spotted onto that. So the company that we get these from has cut up the entire genome into little pieces, spotted it into an array, and we've asked them, hey, put a lot of spots for chromosome 18 so that we can get extremely high resolution. We can tell really precisely where somebody's, what we call breakpoint starts, or where their duplication region starts. Now, so we have this slide, we add the sample DNA, it goes and it finds its matching section. Again, there's a lot of, looking for your matching section here. And I have a couple different images. I then have to run it on a laser scanner, and we get, I never see this, the scanner sees something like this, because the sample DNA has been labeled with a certain color, and the laser measures the intensity of the color, and then can tell what where the duplications and deletions occur. So this is what it looks like by my eye. But this is one of our arrays. You can see, it's one of our slides, excuse me. You can see the 4 different sections. So in this case, I'm able to do 4 people's DNA at one time because there's a special gasket that fits on and keeps it from spilling over. And we get data that looks like, let's see if I'm going to click on the right one. Okay, that's just another image. We get data that lets us, sorry people, I'm getting this.
Well, you know what, while you're getting that slide up, Patty, we did have a question that maybe you can work into your talk, and that is a question from Nathaniel, and he'd like to know if there's any way to test metabolic rates for people with chromosome 18 at our clinical research center, and if so, what does that test entail?
Metabolic rate is not like one thing. I'm going to let Dr. Cody answer that.
No, that's not something we do. There's not any data to suggest that's an essential difference between people with chromosome 18 and other typical people. So it's not something that we focused on.
Is it metabolic rate?
Yeah, I don't know how you do that.
But we don't do that.
Right.
Yeah.
So just to recap, so
But there's no data to suggest that's a fundamental difference.
So some of that was a little difficult to hear, so I'm just going to repeat it for all of our attendees that that we don't have data to suggest that the metabolic rates are different for our chromosome 18 research participants, and so that is not something that we test at our clinical research center.
Right.
Is that right? Okay, great. And we had one other question from Maria, and she would like to know if we can receive information or researches from cases in Europe.
Oh yeah. It depends what it depends what you mean by that. We certainly involve lots of people from Europe, and we can get their information, and we send them the surveys and all, and they're full participants. If you mean that you have to enroll in the study to be a participant, I mean, you have to send the medical records, send the blood, etc. So it doesn't matter where you come from. Can some random person just send us medical records? So, I mean, I don't care whether you live in San Antonio or Europe, you can't just send us your medical records and have it included. You have to be consented, you have to send the medical records, you have to verify it from some abnormality, and then use the blood. So we say process. So I'm not quite sure how they meant the question. That's the process.
Great, thank you for that. And we are, for all of the attendees, we are going to go a little bit more into detail after we finish at the lab about how you can enroll in the study.
Okay, so I have finally pulled up my slide showing where we go from this glass slide that has the 4 microarrays with the invisible to the eye dot that the scanner then sees as a common variations of red, green, or yellow. And then the software analyzes that, and this is an example of the type of data we would get. Each of these is a different study participant, and each of these dots, and you can see maybe these red dots a little better, are one of those 180,000 spots of DNA that's on the array. And then you can kind of ignore the colors. Those are just there for our convenience to see it. But where there are 2 normal chromosomes, they line up right along here because this is a log scale. I'm not going to explain that any further, mainly because I'm not 100% sure how to. And if you have a shift to the right, which you can barely see up at this top area, there's a little shift to the right. That is a duplication. The bottom here, where you have a shift to the left, that is a deletion. And we can zoom in and tell you with a lot of confidence where exactly that breakpoint is and where exactly this duplication point starts. So then, excuse me, over here we have somebody that, you scroll down here, they have a terminal deletion, 18q minus. That would look similar to the fish, that first image I showed you of the chromosomes with the red and green dot. And then again, another person who's got a larger 18q deletion of the telomere. And then here, someone who's got a deletion, because see, there's a shift to the left, but it's interstitial. And
What's
this
interstitial?
interstitial is when it's in the middle of the chromosome or somewhere above. It doesn't involve the telomere. It doesn't involve the end of the chromosome. And this part of the chromosome and this part have actually joined together. So it's like you cut this piece out. Now, microarray is wonderful because we can get these results for the entire genome, and obviously targeting chromosome 18 in a day. The original technique that we were using when I first started working here was PCR and then running that on a gel, and it would take a minimum of 3 days to get information on just one of these spots. And it used radioactivity, which nobody wants to use. So this made my life so wonderful. Now, what microarray tells us is copy number variation. A fancy way of saying, do you have extra? Are you missing something? Okay, it tells us duplication or deletion. It doesn't necessarily tell us where that duplicated material is. And it also doesn't tell us if there's a ring chromosome. So that is where you can also go back to fish, that first technique I showed you. And all I want you to look at on this slide is they've used this turquoise, this aqua color to identify chromosome 18. So here it is. Here's another one. And here's its telomere. And I can't really see, but they enlarged it. This one chromosome 18 has made a ring. It has formed a circle. So this is somebody that has ring 18. We would need this procedure, this technique to identify rings. The microarray can tell us where the deletion is because typically ring 18 involves a deletion, but it doesn't confirm the actual shape or morphology. So we're doing lots of different things to answer big picture. So the final thing I do with the blood is I don't want to have to get blood more than once, and you use up, potentially you could use up the DNA. I don't want to have to, have you guys send more blood. And we might come up with other questions that we need different types of cells. So I establish a cell line. I spin the blood in this kind of tube that's got a gradient, a gel, and it separates out the white cells, which are the ones that contain DNA. I then culture them. I add them to a flask. It already is. Oh, sorry. Leave on.
Patty, while you're getting your glove on, we have a question from Jessica, and she would like to know if you study Pitt Hopkins as well.
Yes, we do. Yeah, one of the chromosome 18 conditions. Yeah, it's just a subset of HTT minus. So yes, we have enrolled people that have that. So what I do, set up the cells. I get the white cells. I actually start in a small flask like this. I can show you guys this one, even though it's not being used. And I add growth media, which is the liquid. I also add a virus that does not affect our studies at all. But what it does is it makes the cells immortalized, because healthy cells from your body won't grow forever in the lab, and we want them to. So I add this virus, and these are cells that grow in suspension. They're floating around. And as they grow and get transformed, we get clumps of cells, which I'm trying to show you guys.
Oh, there we go.
Getting direction from David. So hopefully you can see floating white stuff. It takes about 2 weeks before I can see this by the naked eye. And this is the fun part of working in a lab, because this is really subjective. And Dr. Cody likes to say there's some magic to growing cells, because I start them all off the same, but then some take off faster, and so they get fed on a different schedule. What these would look like normally, if you were here, and hopefully someday you guys will be able to come visit us in person, we would look at them under the microscope, because what they will look like once they've been transformed is similar to this. This is kind of the time frame. At day 1, you just see lots of little individual cells, and they're round and happy. They start to, by day 7 to 10, have started forming these small clusters, which is a sign that yes, they've been transformed, and this is what you want them to do. And that just continues by day 14, by day 21. And so you still have some of the individual little cells, that's fine, but these are the important signs that the cell culture is healthy and growing well. What I typically do now is I freeze the cells. I don't have anything actively requiring them. So once I grow up enough, I spin them and add a freezing solution. So the ice crystals don't form in them, basically. And then David's going to maneuver around.
Oh,
Sorry, I should have had a song and dance here. Actually, I did do a song and dance. You guys didn't. Okay. So I freeze the cells in little vials that are stored either in a cane like this, or in a box. And actually, I typically use a plastic box because they work better, but I don't want to, all my plastic boxes are in the tank. And ideally, I grow up enough cells that I can freeze 3 vials for each study participant. And that's so that we can store them in our 3 different liquid nitrogen tanks. Because these are very expensive thermoses. You guys thought a Yeti was expensive? No. These are way expensive. But they can also just fail. It's like an expensive thermos, but it could break. So we never want every one person's samples all of the same tank. The tanks are filled with liquid nitrogen, which is 300 degrees below zero if you're in Fahrenheit. And then I just transfer them. And they can be stored like this forever. I have thawed cells that were frozen in 1993 and they've grown. So that's pretty phenomenal. I realized when I looked at the website, there is outdated information where you say that it costs $10 a week to keep the liquid nitrogen in one tank. That's actually gone up and it's almost $20. It ranges between $17 and $20 just to maintain per tank per week. So basically $220 a month to maintain them in liquid nitrogen. But that is the way cells can they can be stored that way for longevity. The final thing I'm going to show you is my tissue culture hood. When I'm working with the cells, that needs to be done in a sterile environment. And I don't have the blower motor on because it's so loud you would never hear me. But basically, when the blower motor is on, there is a current of air being blown, and it prevents steam from the outside air getting into there. So anything I work with under here, when the blower motor is on, is sterile. So when I process the blood for cells, when I feed them, when I'm going to manipulate them at all, everything's done under here. This is the growth media. It can only be opened under the hood. Because if I get a single bacteria, which could be floating in the air, we're all a little worried about viruses floating in the air, or fungus, it will grow so much more quickly than the cells that it will kill all the cells. It will use all the nutrients. So this is that's how I do that in the tissue culture hood. And I try to keep minimal stuff in there again, so airflow is great. I think that's it. Thank you very much. If you have any questions, now is your time to ask them.
Thank you so much, Patty. We really appreciate it. We are, for all of our attendees to know, we're going to leave the lab in just a moment and go back to Dr. Cody in her office where she won't have to wear her mask and it'll be easier to hear because this is a research hospital. Everybody's wearing their masks. I mean, we weren't allowed to film without masks, so we had to do it this way, you guys, and we appreciate your patience. And Patty, I heard you promise us a song and dance, and I didn't get to see that. I'm really waiting for that.
I did it so fast. You missed it. So.
Oh, was that it? Okay. All right. Well, thank you, Patty, and we'll see you the next time. And all right. Bye-bye. Okay. Bye-bye. Well, while we are going to transition over to Dr. Cody in her office, where, as I said, she'll be able to talk without her mask. It'll be a lot easier for us to hear her. We have a few questions that she's going to answer. But while we're doing that, we wanted to let you know how you can get involved in the research study. And if you would like to get involved in the research study and you're not already in it, it's free of charge. There is no fee to become involved in the research study. You simply already have to have a chromosome 18 verified diagnosis from a genetic test from your own medical records. And you would reach out to Annis Hill. We're going to have her email posted in the chat box. You would reach out to her and she'll let you know about how we get started with consent forms and how we go about, we send you a blood collection kit, you take it to your local lab, you get a blood test done and you send it to us. So she would be able to answer all of your questions about that. We have about 700 participants in the study right now. We can always use more. The more participants that we have, the more valid the research becomes, the more broad the findings can be. So we're very excited to get as many participants as we can. So if that's something that you're interested in, please send Annis an email and she will get you started on, and she can answer any questions that you have. I also wanted to point out that for each participant that we have, as I said, there's no fee, but you saw in Patty's lab, even just a little lab, what do you call that, slide? I'm not the medical person, as everyone knows,
Microarray
I'm the
slide.
people person. Yes. So anyway, okay, Dr. Cody's with us, so I'm going to turn it back over to her. Go ahead.
I guess I'm here for questions at this point. This is back in my office where I spend an awful lot of time.
Yeah, we had a couple of questions that we wanted to hit, and one of those is what does this science mean for families?
Well, it slowly evolves, of course, over time, and at the moment, I mean, we have syndrome descriptions and physician management guides that are all much more detailed, and we have more information about what medicines people are on, what works, what doesn't work, what conditions they have. We are always working to update those. We're looking for now, it's sort of generally described at this point, and so we're interested in sort of the more hidden things like the platelet problems or the bone density problems, that kind of thing.
And what will that mean for families when you find those answers?
Pardon?
And what will that mean for families when you find those answers?
Well, obviously, then they can act on them. I mean, if you find out that you know, your child is at risk for low bone mineral density, then you need to talk to your doctor about what you do about that. And we'll have, of course, direction for the doctor. So all of it is improving health and hopefully, therefore, improving their life.
Great. And Jacqueline wanted to know, do you have to live in Texas to be enrolled in the study, or can you live anywhere?
No, you remember the map? People came from everywhere. They're from all over the world. You just have to be able to speak English and understand the consent forms. That's it. And get blood to us within 3 days.
Exactly. Great. And Nathaniel would like to know, how many people are currently enrolled in the 18Q- study?
That was in a slide in the research update. 350, something like that.
Okay, great.
Getting close to 400.
Very
And
good.
those are family numbers. So we have some families with more than one person. So it's really more than whenever you see the number of enrollment numbers, each number is a family. So it's often just a little bit higher than that number.
Great, great. And Nathaniel would like to know what is currently funded at the Chromosome 18 Research Center. So what studies are being done?
Right now we're just hoping to be here by the end of the year because the registry events are all canceled. And so that's what funds what we do. And so at the moment, what we do, what I consider our sort of baseline level, is keeping everyone employed. I mean, when your newest members of your group have been here 13 years, they know an awful lot about the chromosome 18 abnormalities. That means everybody else, Patty, David, all the others have been here even longer. And so they are really experts in what they do. And they're actually very committed to all of you. And so what we can still enroll families. We still do that as sort of our baseline thing. Still keep collecting medical records, keep asking people for more medical records as their children get older. And we can send out surveys, and they're not, and often they're validated instruments. It's not just Janine making up questions to ask you. They're real
validated instruments so we can demonstrate a behavior or a history of something or whatever. And so those are the things that are ongoing all the time regardless. So did that answer your question?
Yeah, well, hopefully so, that answers the question. And a lot of these questions, some of these questions were coming in as we went along, but we were trying to save some of them until it was easier to hear you.
Sure.
So Sarah wanted to share, in reference to the other question that you just answered, Sarah wanted to share that for their family, it meant the research meant becoming aware of issues and being able to follow up sooner, and that's very helpful.
Good. Yay.
Yes.
Thank you for that answer.
Yeah. Let's see. I think we have, Nathaniel has one more question, but Nathaniel, I'm going to save that one for our Executive Director, Neil. And I did want to ask also, what would you do, Dr. Cody, if you had more funding? What's sort of your wish list? What would you want to do?
Oh, it's long.
Well, give us a couple.
But to kind of pick out like two main categories. One is, there's still a lot to learn from clinical evaluations of people, and especially people with unique deletions or unique duplications, or really small deletions or duplications. For a lot of the features, we don't know which gene is responsible, but we've narrowed it down to a group of 10 genes or some small region. And sometimes we get a participant that just has a part of that region missing, and we'd love to evaluate them. So more money to bring in people to do clinical evaluations. We do as much remotely as possible. In fact, we're working on a project that uses a Fitbit, Apple Watch sort of device, but it gives us numerical data, not just cute graphs. It gives us numerical data we can analyze, and we're going to start, we're testing, beta testing among our group now the, let's see, third or fourth device that we've tried out over these many years. They keep evolving so fast to get clinical information. But so anyway, that more clinical information is one part, but the other part is now we're understanding which are some of the key genes, and we need to do some of the basic science to understand how a deletion of that gene affects cell growth in the appropriate cell types, how when you delete that or duplicate that gene in a mouse, how does that affect their body, their behavior. And those are things we need to understand, because once they allow you to develop assays to test different compounds for drug treatment. And so first, you use different compounds in screens using cells in a culture dish. And when you find something that works, then you move it to a mouse. And then, so anyway, that kind of work costs a lot of money as well. And so we're stuck. We have the first steps on a couple of those, but in the mouse study, the mouse sperm are frozen away because we can't afford to bring back the colony. We don't have the money to house the mice any longer, and cells are frozen away because for another study, because we don't have the money to proceed to work on exactly understanding the differences in the sort of, in this case, 18q minus type cells of the brain.
Well, that's, I love hearing about all this
science in the clinical.
Yes. Well, it sounds like there is a long list, like you said, a long wish list of things, and there would be no shortage of ways to spend money on research in ways that are really wonderful and cutting edge. Love the idea of those Fitbits. That would be really great. So, all right. Well, Dr. Cody, one other question I wanted to ask you. When people go on the internet and they search for chromosome 18 information, and if they find it on Wikipedia or they find it wherever, where did it usually come from?
Oh, that comes from us. I mean, we wrote the Wikipedia, what is it, GARD, the genetic and rare disease, whatever. We either wrote, I say we, Courtney, either wrote or we edited that. Genetics, what is it, genetics home reference or something like that. I think we wrote that. And for the doctors, there's a service called Up to Date that is linked to medical records. So a doctor in the hospital or in their practice, if they subscribe to Up to Date, right within the electronic medical record, they can click on Up to Date and go to look up the condition the patient has that's sitting in front of them. And I wrote that information for chromosome 18 abnormalities for up to date. So it cracks me up when somebody, gives a talk and says, oh, here's some references. And I think they're all secondary references that we wrote.
Yes. Well,
whatever.
not only are you doing,
but I can't guarantee what everything you find on the internet. So when we find something out there, we send people edits and corrections, and usually they thank us profusely.
Right.
So I can't guarantee if it's not on one of our websites, either the Chromosome 18 registry or the Chromosome 18 clinical research website. It's not on one of those. I can't guarantee that we edit it for accuracy, but we try.
Very good. So you're doing the research and you're getting it out there all over the place to make it as accessible as possible.
Yeah.
Like Dr. Cody said, please check our website, thechromosome18.org, or the registry the Clinical Research Center website, which is in your chat box right now. So we really appreciate the tour. Thank you so much. We're gonna let you take a breather now and say goodbye to you, and we're gonna kick it back to our executive director, Neil. Thank you so much, Dr. Cody. We appreciate it.
Thank you all for being here.
Bye-bye. All right, and Neil, you were going to give us an update on our events, and if you can also work into your talk, we had a question from Nathaniel about where do we get our funding?
Yes, kind of great questions, and thank you everybody for hosting the questions. I'd also like to say a big thank you to Dr. Cody and the team over there. Is that obviously, this is all new to them, and it's all new to us as well. And it takes a lot of time and effort to kind of put on something like this.
The touring is new, but they're pros at research.
They're pros. They're definitely pros at research, exactly. And want to also give a very quick shout out to our Director of Operations, Amy Behan, and of course Michelle for kind of helping us host as well. And our intern as well, Anya. So thank you very much indeed for everybody to kind of help put this all together. But it's been a very difficult year, as you can see everybody wearing their masks. We love to host the in-person tours, and like every other event, kind of throughout most of the country and internationally, is that we've had to cancel all of our events through the year, which includes our conference, it includes our kind of big 5K run walk, golf tournament, and it's made a big effect on us financially. We really hope that our families value what we do, and part of why we want to, kind of show everybody that some people forget that every day our team is working hard and diligently to make sure that we're providing as much information as possible. And whether that's in kind of the medical guides that people can give their physicians, or whether it's some of the new projects that Dr. Cody was talking about, is that we're working every day to kind of help improve the lives of kind of kids and families around the world. And as we continue to do that, is that, we've tried to put some events virtually. We have put events virtually online. One of the things we're running at the moment is the Power of 18, which is, Michelle might talk a little bit about at the end. But, it's a way of being able to help in a very small way. But if we get enough people involved, it can help in a big, big way. So we're kind of very excited about that. In November, we're going to be having a virtual 5K run walk because we can't have our in-person. So hopefully people can get involved in that in their neighborhood, get neighbors, friends, family involved. They don't, they can all do it remotely, so they don't have to be together. But it's a way of keeping the funding moving through. And at the moment, about 80 to 85% of our funding comes directly from our families. We do reach out to foundations, and we have some wonderful corporate sponsors as well. But again, most of those are all built around the event. So as we lose those a little bit, is that, we're really asking our families to kind of step up and help us kind of keep things funded to where we need to be. Did you have any other questions, Michelle?
You were hitting it. Sarah was asking about other funding sources, and you were talking about how much comes from our families. Can you elaborate just a little bit on our community partners and how you go about raising awareness with them?
Yeah, so you know, kind of how can people help? Is that, let's kind of talk a little bit about that. Is that, you know, there's some amazing stories about how our families have got involved that go from the bizarre, which was, I think the most unusual thing I heard was when one of our members auctioned off a goat in California, and their friends and family got around and they supported them by paying $30,000 for a goat. Is that, you know, going to do talks at schools and raising awareness. We had one high school student that talked about her sibling and did it to a bunch of parents, and somebody in the audience, you know, was touched enough that they donated $30,000. Now, you know, a lot of it is about the money, but so much of it is also about raising awareness with other people in your community that can help us, you know, kind of help us raise awareness about what chromosome 18 is, and kind of what the community means to everybody. So, you know, you can volunteer online. We couldn't run the organization with all our wonderful volunteers. You know, whether that's the board of directors, you've got a few hours a week that you can help us doing some social media posts, or you've got a high school during the summer that might like to kind of have some projects or what have you. You know, we can work with that. So is that it's really a question of getting engaged, but kind of coming down to other areas of support from the foundation. We have different foundations that we reach out to. We had a wonderful, you know, we have people like the Baptist Healthcare Foundation of San Antonio, which is actually helping support us in a small way with regards to funding the watches or the kind of the devices that Janine was talking about. So, you know, so we then have corporate partners. And as an example is that we have the Batello Lumber Company that has been running golf tournaments for us for actually it was going to be their 20th anniversary this year. And they've raised, I believe, almost $500,000 over that 20 years to help support us. So it doesn't have to be that big. Is that we also then have people that do lemonade stands. We have people that, you know, kind of do mountain bike rides, or it can be Pokemon, you know, hunts, or, you know, really the world's the oyster on that. But still the majority of our funding, between 80 to 85% does come from our families.
All right, very good. And what about, can you talk briefly, we're about out of time, but could you just briefly talk about the grants that that we work so hard to apply for?
Sure. Well, we have a volunteer grants team, which we have either as interns, or we have some work experience with different universities, or actually during the summer we actually work with some high school students as well. And what we do is that we reach out to a network of foundations that we've built up over a time. A lot of them actually based here in San Antonio or Texas, but we do also reach out to national foundations as well to kind of raise awareness about what we're doing. We kind of go through the grant process. This can be a very long process sometime. For example, next year's Baptist Foundation grant we actually started writing the grant, I believe, in March, and we won't hear until November. So, but you know, some grants will kind of come through straight away. We did apply for some the PPP loans, which for anybody in the US was the assistance for not-for-profits and small businesses. So we were lucky enough to get a small amount
Because
for
of
that.
COVID.
Yeah, for COVID. We actually got a $25,000 grant from the San Antonio Area Foundation for something specifically with regards to help get the information and messaging out to our members about, you know, additional COVID support as well. So it's a very wide net. So if you've got any connections with foundations or corporate partners that you can introduce us to, it always helps a huge amount if we have that personal introduction.
Yes, very good. Thank you, Neil. And wanted to clarify, part of the reason that Neil was saying that we have some regional grants that we apply for, some things you have to be located in the state, or I'm not as familiar with that as you are, but just wanted to clarify that,
Yeah,
it wasn't
so,
for not wanting to apply for all over.
No, believe me, if we had the time and the people power to do it, we would apply for as many grants as we could, but some are very specific geographically to where you have to have an immediate effect in an area. And then you have kind of like, you know, kind of bigger foundations like the Bill and Melinda Gates Foundation that you have to be invited to apply for an application. So every process is different, but we're always, you know, always ready to look at any new opportunity.
And always, as Nathaniel pointed out in the chat box, as our wonderful executive director Neil pointed out, is to raise awareness right where you're at. You would not be on this tour if you didn't care and have a vested interest in chromosome 18. So letting people know by wearing a hat or wearing a shirt, which are all for sale in our merchandise, can raise awareness. Letting people know if you're involved in Power of 18, posting it on your Facebook page along with your story, and then people will know. So thank you so much for all of that wonderful information, Neil. We appreciate it. So we're going to wind down. I'm going to give just a few, thank you, Neil. I'm going to give just a few announcements before we end, and then we'll be on our way. I wanted to let you know that, let's see, we talked about our merchandise, which this year is our 30th anniversary. So right now you can get the limited edition 30th anniversary. You can select either 30th anniversary or just the regular chromosome, I can't even talk, chromosome 18 logo. I got both, so I wanted to have both. So we also have our online auction going on. There's a beautiful Coach bag there that I have my eye on. You can bid on those things now. Oh, wanted to mention our merchandise. We are limited to US only that we can ship to, just because there's laws and things governing where you can sell what types of things. But you can get in touch with one of your Facebook friends in the US and maybe ask them to help you out, maybe buy something and ship it to you. But we can't do things like that. In fact, we didn't even say that. But wanted to remind you that all of our in-person fundraising events have been canceled. So if you can support us with online auction, Power of 18, and our merchandise sales, that would be amazing. As always, you can go directly to our website, chromosome18.org, and make a direct donation in the name of the person that you care about who is affected by chromosome 18. I wanted to remind you the recordings of all of our events are available on our conference website, except for our Club 18 and our sibling panels. Speaking of our sibling panel, that's coming up this afternoon at 3 o'clock Central Time in the US. That's all of our wonderful siblings that will get together, answer questions. It's really insightful and a wonderful time. So if you are a sibling, make sure you are there. And we also have today sessions on allergies and autism. So you can look for those links on our website. Coming up tomorrow, we have neurology with Dr. Atkinson, and we have ENT talk with Dr. Perry. So we will see you all then. Thanks for coming. Bye-bye.